So hello everyone, I hope you can hear us. We are now presenting the first webinar that's jointly organized by the Journal of Parkinson's Disease, the Cure Parkinson's Trust, and Parkinson's Movement. And this particular webinar is focused on the effects that exenatide might have on non-motor symptoms in Parkinson's disease. And it's based on a paper that was recently published in the Journal of Parkinson's Disease, where there was a post hoc analysis done on the data of an original study which was published last year. So I hope you can all hear us, and I would like to introduce you to the panelists. First of all, I am Patrick Brunden myself. I edit the Journal of Parkinson's Disease and have been doing Parkinson's research for almost 40 years, and I'm sitting in Grand Rapids, Michigan. Our next participant, who could perhaps wave, is Professor Tom Faltony. He was is the lead investigator on the trial with exenatide in Parkinson's disease, and Tom is located at University College London. And if you go down to the next screen, if we could have a little wave from Nigel Gregg and Dr. Dimitris Kapogiannis. They're both at the National Institutes of Health here in the United States and have a long background of research in mechanisms related to the effects of exenatide in the body. And we'll be discussing some of this as it pertains to non-motor symptoms and potentially slowing disease progression. And finally, in the best location of all, at the bottom of our screen, we have Penny Custo, who is a participant in the trial, or was a participant. And Penny is located in a caravan in Cornwall. And you can see through the window there, it's quite beautiful outside. So those are all the panelists. I'll just give you some ideas about what you can do if you are listening in. It's possible for you to send us questions on the Q&A window, which you will find either at the bottom or the top of your screen, depending on what type of computer you have. And if we see questions come up during the discussion, I will try to incorporate them. I should also tell you that we've already received a good number of, I would say, about 30 questions. They're actually clustered around about 4 or 5 themes. So you may hear your question, but it won't be exactly worded the way that you sent it in, perhaps, because I'm going to try to group them. And those questions will be introduced to the webinar as we move along. We are planning to discuss the following themes, just so you know what's going to happen during the next hour. First, Tom Faltony will describe the findings in the original Lancet paper that came out in August last year, which was the exhaustive report on the findings in people with Parkinson's disease who were treated with exenatide in a blinded placebo-controlled trial. Then Penny will discuss her own experience of non-motor symptoms and whether or not they seem to change during the trial and how difficult it might be to track these symptoms and how they will vary between people with Parkinson's and how they can very significantly impact quality of life. We will then let Tom come back and describe this most recent paper where they revisited the original trial and the team of investigators looked specifically at non-motor symptoms. As we hear those findings, Nigel and Dimitrios will be getting ready to discuss how exenatide actually might, at the molecular level, impact non-motor symptoms or other symptoms in Parkinson's disease. This is a very, very exciting area where there have been publications, at least one major publication coming out just about 2 weeks ago, or less than 2 weeks ago. And we will probably sort of wrap up with Tom discussing the future outlook for exenatide in Parkinson's. Will there be another trial? Hopefully there will, I can say, but what type of trial and where are the plans right now? And that's basically it. So I want to kick off by asking Tom Faltony, please, to describe the initial paper, the Lancet paper that came out in August last year. What were the findings?
Thanks very much, Patrick. It's a pleasure to be here and to have the opportunity to discuss this. So we started this work some years ago and it all came out in the public arena last August. And this was the report of a trial of 62 patients with Parkinson's disease who had all already started treatment with levodopa. And we randomly allocated the 62 people into two groups. So this is a randomized controlled trial. And half of the participants were randomized to inject themselves once a week with an active drug called exenatide. And the other half, again, the randomized group were randomized to inject themselves once a week with a placebo. So the participants had no idea whether they were injecting themselves with the active drug or the placebo. And this is what is meant by a double-blind placebo-controlled trial. The participants didn't know what they were injecting, and the people who were rating their Parkinson's disease severity during the course of the trial also didn't know who was injecting placebo and who was injecting the active exenatide. So all the participants continued to inject themselves for 48 weeks. So that's 48 single injections. And then they stopped the injections and we saw them again 12 weeks later. So during the course of this 48 or 60-week period in total, we did assessments every 12 weeks using a scale called the Unified Parkinson's Disease Rating Scale, which is the standard, the gold standard way of measuring Parkinson's disease severity. But it very much focuses on the movement problems of Parkinson's disease. This is the tremor and the slowness and the stiffness of Parkinson's disease. And because they were already on levodopa, what we asked people to do was to attend those visits first thing in the morning before they'd taken any of their dopaminergic medication. So we got a chance to see what their Parkinson's was like in the absence of that noisy signal from the different doses of levodopa. So during the period of the trial, we could get some objective measure of Parkinson's disease severity in both groups and see how it changed during the course of the follow-up. And what we found is that those patients that were using placebo, so the 30-odd patients on placebo, had a gradual deterioration in the motor severity of their Parkinson's disease during the 48 weeks that they were injecting the drug or the placebo, and that continued the 12 weeks afterwards. So at the very end, at the 60-week time point, the placebo group were worse. The patients that were on exenatide during the period of the 48-week treatment exposure, and then even beyond that to the 60-week point where there's no longer any of the exenatide left, were remarkably stable. If anything, there was a very modest improvement in the severity, the motor severity of their Parkinson's disease throughout this period of time. So we reported this in the publication, and this was our primary outcome that we decided before the start that we wanted to see if this drug had an impact on the motor severity, and we found that it did. So this was statistically significant, and it was an important finding because even though this advantage was small, we were looking to see if this drug may do something to perhaps slow down the rate of progression of the disease. And this was the first double-blind placebo-controlled trial that confirmed that this certainly appears to be the case. So that's what was published in the Lancet. What we also published were a number of what we call secondary outcome measures, where we looked at non-motor symptoms, we looked at cognitive performance, we looked at quality of life, and in all of those comparisons, there was a minor hint that exenatide was better, but there was nothing that reached statistical significance. And so that was, if anything, it was a little bit disappointing that only the primary outcome was the clearly positive signal. So from that point, we had published the data, and we had a very large data set to be able to scrutinize further. And we decided to do more of a deep dive into the data and try and find out, well, is there any signal above and beyond what we'd reported in the Lancet paper?
So before you go there, Tom, could I ask Penny maybe to tell us a little bit about non-motor symptoms and how they typically impact the lives of people with Parkinson's? And perhaps share some of your experiences in the trial.
Yep, of course you can. I was a very willing participant in the trial because I felt that it was something that I was perfectly suited to. I've had PD I was diagnosed at that point, I think, by 5 years. It's now 6.5 years. And I don't consider myself as having major symptoms to notice. You would notice it if you saw me in the street. But behind the scenes, there's loads going on. As you know, all the common ones: anxiety, overactive bladder, dry mouth, overheating. I get restless legs, I get cramps. I think my cognitive skills are okay, but I can't concentrate for as long as I used to on any one thing. I jump. Obviously, the first thing was the lack of sense of smell, which probably predated Parkinson's by 5 years. And my sleep pattern's disturbed, handwriting's tiny, all the things that everybody talks about, none of which separately sound very drastic, but when they all come together or come one after the other, it's not good fun. The Exena trial was only a trial for me, because being off levodopa rather, is trying, it's difficult. You have to be off it, I think, 36 hours. I don't think it's just overnight, Tom. I'm sure you made me be off it 36 hours, and boy, was I weakened by that. But I changed quickly. Once the levodopa's in, it changes you. And any of the tests that I did for Dylan when I went back and forward to the hospital, I felt confident that I was performing the same as I had last time. I didn't notice any deterioration at all. I don't think I noticed improvement, but I certainly didn't deteriorate. And that was, for me, was very hopeful. And of course, as a result of it, I've found out I was on it. And luckily, I'm still on it. So I'm taking it as it comes.
So before I actually ask Tom then to comment on the results of the post hoc analysis on non-motor symptoms, I realized I forgot to tell you that I have to read our conflicts of interest, and I apologize for not doing that. So Nigel Gregg, who's participating in the panel, is actually named as an inventor on the patents that cover the use of the class of drugs that exenatide is part of, the GLP-1 receptor agonists. This is work he's done over many years, starting a long time ago, in several neurological disorders. But the patent rights are assigned to the US government and the nation of the United States. But he is the inventor. So that can be perceived as a conflict of interest. And we should say, of course, Tom Faltony is the lead investigator on the trial that we are discussing now. So that can also be perceived as a conflict of interest as one describes the results. So with that out of the way, Tom, could you please describe then what was the post hoc analysis of the non-motor symptoms in people like Penny, who was treated with exenatide in the trial?
So when you're left with a very large data set, even though you've described the a priori, the original hypothesis, and described the results of testing that hypothesis, there's a whole load of data that is still there that's only been partially examined. And so this is what we call about doing a deep dive. Other people will call it data dredging. It's the same thing. It's just trying to see what other messages might be learnt from looking at all these different data sets. And within it, we had a whole lot of detailed information from the non-motor symptoms scale. And this is a scale that asks people to rate the severity and the frequency of symptoms related to mood or anxiety, but also related to bladder and bowel, related to sleep, related to sweating, or even sexual function. And so all of these composite symptoms are lumped together, and the result of that means that you can gauge what change there is in non-motor symptoms in their entirety. But it doesn't tell you whether any subsections of that scale may have changed if you just look at the round number at the bottom. So what we decided to do is to break down that scale as and I have to emphasize this, a post hoc analysis, that we wanted to look to see if there's any evidence from this data that something might have changed. But it's not a way of robustly deciding if we can have strong evidence of change. It's just a way of generating a new hypothesis that we may be able to test in the future. So I start off by saying this is a post hoc analysis, and there's no way we should use these data to make a definitive decision, but it's a good way of generating new ideas. So in this post-hoc analysis, we look to see if the depression components of the non-motor symptom scale, if they changed between people on placebo or exenatide, and we found that there was a consistent change that favoured patients on exenatide. We also looked to see if other items that captured depression changed in the same direction favouring exenatide, and we used the very first part of the UPDRS, which asks about depression, and also the quality of life scale. There's an aspect in there that asks about emotional well-being, as well as the depression scale that we used in the original trial. And by carefully going through that and making adjustments for differences that occurred at baseline, we were able to consistently find that patients on exenatide did have an advantage compared to patients on placebo. So those data should be viewed as hypothesis generating. There's a signal that perhaps depression might be a little bit better in patients on exenatide, but we won't know with any certainty about that unless we take that hypothesis forward into the next trial and see if it stands up to and is consistent. So that's what we've learned from this latest deep dive into the data.
So I just welcome Penny back. I think you dealt with some overanalytic results or something outside your caravan. Tom, there is a question that's come in, I think, specifically to you. If the objective is to modify disease, halting or slowing progression, then surely if non-motor symptoms don't decline, wasn't it a success? Can you explain sort of the intricacies of why you can't claim an absolute success, but you're only generating a hypothesis? Can you just give a little bit of a sort of layman's background to this?
Sure. So firstly, this is a post-hoc analysis. So no matter how strong the statistical...
But explain the word post-hoc here. So what does that mean to the...
Right. So post-hoc is just the Latin for after the event. So we've decided to do an experiment. We've done the experiment, and we've decided how we were going to look at the results beforehand. And so those results are robust and valid. But if you decide to look at the results in a different way after the event, then you can't be as confident in those results as if you decided to look at that way of doing the analysis beforehand. And that's just good science, that you shouldn't do a post-hoc analysis unless you're willing to have a degree of skepticism about the results, and you use them for hypothesis generation only. And that's the most important message to get out to people, that a post-hoc analysis is good for planning the next trial, but shouldn't influence the decisions that we're making clinically today.
And couldn't we also say for the lay audience that if you look at the same group of people in the same trial in 100 different ways, by chance, a few of those ways that you look at them will show differences, even though there are not true differences in the outcome. So that's the danger of testing too many things on the same group of people.
That's absolutely right. So the statistics, are there to help us, but but shouldn't be overinterpreted.
This time, it's a good idea.
We find differences in the primary outcome.
Oh, sorry, Tom, you were breaking up. You were sounding like a Dalek from Doctor Who. So I don't know if you have a bad line, but I think it's a good time to arrest your Wi-Fi or your internet and ask Nigel and maybe Dimitrios together what could be the mechanisms. Now we're speculating because, as Tom said, we're really only looking at hypotheses that were generated from this data set. What could be mechanisms underlying the possible effect on non-motor symptoms?
So I will just start by saying that the issue of addressing mechanisms clinical trial is related with the issue of biomarkers that show response, therapeutic response to the intervention. So the field of biomarkers is something that it is currently, research is expanding and focusing exactly for the reason of providing approximate outcomes to researchers that are interested not only whether at the bottom line, whether a medication worked or not, but by which mechanism it did work if it didn't, and trying to generate knowledge that it is generalizable about a mechanism of action in a disease rather than only one particular agent. Because if a drug works in a particular trial, we don't know if that would mean that any drug that shares the same mechanism would work or not. These are questions that you can only answer if you have therapeutic outcome biomarkers. And I will let perhaps Nigel take the lead. What mechanisms at the cellular level you would expect exenatide to have an action on?
Okay, so I think that was very useful. And because as Tom's indicated, the clinical measures that he's looking at in his disease, which relate to either motor or non-motor, they're clinical measures, and any underpinning mechanisms that could impact those clinical measures, they could be one mechanism or they could be numerous mechanisms. And one would then have to follow particular markers of those mechanisms in the people. And that's what Demetrius is going to talk about. And when one considers mechanisms, it really depends on how you consider Parkinson's disease. Is there a primary mechanism, or is there a primary, conceptual pathogenic mechanism that underpins Parkinson's disease, or are there numerous ones? And likely there are numerous ones, and those numerous ones are slightly different in different people. And I think exenatide is an interesting drug because it doesn't really work through a single pathogenic mechanism. It impacts many processes that are involved in the optimization of the normal brain, the normal healthy brain, and it also impacts a number of things which can go awry in a pathological process in Parkinson's disease, and many of those pathological processes are across diseases. Some are neurological disorders, such as this drug's being looked at a number of things, Alzheimer's disease as well, but some of them are non-neurological disorders, such as type 2 diabetes, where this drug was developed in the first place because underpinning mechanisms there have some parallels in the brain. So how does this drug potentially work? And why does it work in this case in Parkinson's disease? And I think Parkinson's disease is different, as Tom's indicated, is that there's a pathological process where dopaminergic neurons are primarily impaired, but there are other neuronal systems in Parkinson's disease that are impaired as well. And many of the non-motor symptoms may be due to those dopaminergic neurons and then secondary systems that they impact, or they could be due to other systems, other neuronal systems in the brain getting impaired. But primarily it's a dopaminergic system. The dopaminergic system is different because it's one of the systems that is that has huge connections, and its sort of arborization, or the number of synapses that come from a dopaminergic neuron, is just phenomenal. And they're primarily unmyelinated, but it's considered in the human brain that one dopaminergic neuron will synapse maybe 1 million to 2 million other synapses. So these neurons are kind of, under normal circumstances, they're working very, very hard. In the aging process, they get stretched. And that's potentially what underpins Parkinson's disease from a number of points of view. So there's clearly oxidative stress that occurs with those neurons. And that oxidative stress, there are a number of pathways which have been looked at with exenatide, with this GLP-1 agonist, where you can mitigate oxidative stress. there are direct actions which are considered neuroprotective actions, where you can mitigate a number of toxic stresses, such as toxins, direct effect of toxins on neurons. Also, neurons survive because they have a whole series of trophic factors that support them. And we know in Parkinson's disease that many of these trophic factors have withdrawn. Many of the trophic factors that are supporting them are actually relate to insulin and the insulin pathways. And we know that in Parkinson's disease, there seems to be some insulin resistance that occurs into the brain. So exenatide is, of course, used in type 2 diabetes. And many of the effectors that are impacted by insulin and insulin signaling pathways, exenatide can interact with those downstream effectors through different pathways, because some of those insulin resistance ones are blocked because of the insulin resistance.
So could I just interject a question there, Nigel? So there was a paper that just came out a week ago, and we distributed it among ourselves yesterday, I think, suggesting that people who have type 2 diabetes onset when they're relatively young have, if I remember rightly, about a 3.8-fold increased risk of developing Parkinson's later in life. As you hinted, there have been links between type 2 diabetes and Parkinson's for many years, but isn't this quite a stunning observation? And doesn't that actually add some sort of weight to the notion that one should look at glucose metabolism, insulin signaling in Parkinson's even more carefully?
Absolutely, you know, and many people, including yourself, are looking at those. And so I think that there is a certainly in Parkinson's disease, because of the energetic load that dopaminergic cells are under, you know, with this huge number of arborization, with these huge number of synapses, is that they're particularly vulnerable to to changes, to small subtle changes. And so for any of these processes that can go a little bit awry over a long period of time, you can well imagine how vulnerable these particular cells can be. So I think you're right, is that many of the signaling pathways, we know that exenatide is actually a useful treatment for type 2 diabetes. Not only does it seem to overcome some of the resistance that is developed in type 2 diabetes, but one of the interesting mechanisms of actions that it has is it has effects on survival mechanisms within cells. And so for pancreatic beta cells in type 2 diabetes, it mitigates the cell loss, the death that occurs in those beta cells. And those same biochemical cascades are available in neuronal cells. And so some of that anti-apoptotic, it's called, or improving the blocking cell death, improving cell survival mechanisms have been looked at, and they have parallels in neuronal cells. So it's then been found, as you've indicated, that not only seems to be an increase in insulin resistance in type 2 diabetes in Parkinson's disease, but you'll also look across other neurological disorders. A recent paper on multiple system atrophy by colleagues of ours, which came out last year by Fair Basil, indicated that there's insulin resistance in multiple system atrophy, which has some parallels to Parkinson's disease, but it affects more neurotransmitter systems within the brain. Demetrius's work has found corollaries in Alzheimer's disease, with insulin resistors and markers of insulin resistance in Alzheimer's disease. And as a consequence, many of the tools that he was developing for Alzheimer's disease, as he'll tell you, he's been able to shift and by collaborating with Tom, to be able to look at these kinds of things in Parkinson's disease.
So I'd love to hear more from Demetrius about these tools, because I know he has some really interesting results and innovative ways of measuring. But just before then, I'd just like you to finish off, Nigel, by saying something about the cells that aren't nerve cells in the brain, because we always talk about the dopamine neurons and other neurons, but there are many supporting cells in the brain. We know of the microglia, which are the immune cells of the brain that monitor immune events and generate inflammation. But we also know of the star-shaped cells called the astroglia in a fancy neuroscientist's dictionary. Yes. There was a paper about a week ago that revealed some really, in my mind, rather unexpected results regarding the role of microglia and astroglia in a Parkinson model where the animals had been treated with an exenatide-like drug. So can you describe this, and what's your take on this?
So one of the key pathological pieces that you look for in Parkinson's disease is the presence of inclusion deposits of a protein called alpha-synuclein. And the structure of alpha-synuclein has been known for some time. And it's a protein that doesn't really have a defined protein structure, but under certain circumstances, it can then form a defined protein structure, which tends to aggregate. And the function of alpha-synuclein seems to be involved in synaptic transmission and a whole variety of things. It's quite an important protein, not only in dopaminergic cells, but other cells as well. And these inclusion deposits appear in Parkinson's disease, as well as other diseases with alpha-synuclein, which are called synucleinopathies. And so this paper, which was an interesting paper, and this had been demonstrated before, but alpha-synuclein, when it aggregates into the brain, and if it's not removed by a process, which is a fundamental process in the brain, it can cause neuroinflammation. And the process of neuroinflammation is actually induced by immune cells within the brain, which are called microglial cells, as you've just mentioned. And so it was found that GLP-1 receptor seems to be expressed not only on neurons in the brain, but also on microglial cells in the brain, as well as the supporting astrocytes. And there are lots of astrocytes in the brain, and they provide us a support structure to optimize the action of the neurons that are present. So each of these brain pieces, it's not just neurons in the brain that are important, but it's each of these pieces working optimally. And so it had been found previously that, that neuroinflammation can impair and drive alpha-synuclein aggregation. And all these processes are linked. There are circles going, it's like spaghetti. There are circles, and you move one piece, everything else moves as well. So what happens is that through neuroinflammation, through oxidative stress, through cells dying, you can induce neuroinflammation. The key thing is that these microglial cells become inflamed. They start to pump out pathological amounts of cytokines, which are pro-inflammatory factors. And so this new paper demonstrated that in a particular animal model using alpha-synuclein, that these cytokines will then induce the astrocytes, which are normally supportive cells, to become unsupportive in structure. And that three particular cytokines, TNF-alpha, IL-1 alpha, and a C1q, which is a complement piece, will cause the activation of these astrocytes into a shall we say, a violent form, which is called an A1 to an A2 form, and that they will then pump out a secondary substance which wasn't identified, that can then impact neurons and cause those neurons, dopaminergic neurons in this case, in this particular paper, to become dysfunctional and then to die. And that exenatide is able to mitigate that response. It will turn off the microglial cells so that they don't pump out these factors, which then initiate this process through the astrocytes. So at many different levels, there are a whole variety of things that exenatide can do. And it's likely doing multiple things at one time. And so it has what I consider to be the sort of pleiotropic actions. Through a very selective receptor, it has a series of potentially useful actions. And it's likely that some of those actions will be valuable in Penny at her particular time, and some of those mechanisms may not be involved in Penny's Parkinson's disease, but may be involved in somebody else's Parkinson's disease. And I think that that's why exenatide potentially has interesting actions, because it has a series of actions which are across, there are neuroprotective actions, neurotrophic actions, actions on neuroinflammation, actions probably on mitochondria and mitochondrial function, etc., and a whole series of additional actions where these will be useful across a number of patients. And then the clinical outcome scores that Tom's looking at can then, because there are a whole variety of actions, can then be improved potentially. And then the mechanisms that underpin these individual actions have the potential to be looked at by looking at key pathways that drive these mechanisms that Demetrius can then evaluate.
So I'd like to just interject. So I'm getting questions on the screen as we're talking, and we will come back towards the end of this presentation to the possibility that exenatide could be interesting in other neurodegenerative diseases. You mentioned multiple system atrophy, and we know there are trials going on in Alzheimer's. And the fact that neuroinflammation is a part of several neurodegenerative diseases makes, I think, this recent paper very interesting, suggesting that exenatide affects that process. But if I'm going to hand over to you, Demetrius, a biomarker for the layperson, it's if you measure the temperature when you have the flu and you have fever, that's a biomarker. If you want to know if you're pregnant or not, you do one of those sex hormone tests, and that's a biomarker. Or if you have diabetes, measure blood sugar. But in the brain, it's not that simple. You can't stick a thermometer inside your brain and monitor things that are going on there. So you have a really innovative way of approaching this. So tell us about it.
These were some great biomarkers, by the way, that you mentioned, and no wonder they have been used for a long time in medicine. But for the brain, in the absence of being able actually to take tissue and looking under the microscope, which for obvious reasons is not an option, we are trying to develop something else that it's being referred to as liquid biopsy in quotation marks. And so what we're trying to do is to isolate small particles called extracellular vesicles or exosomes from the circulation, from the blood, which we think come from neurons or astrocytes, for that matter, from particular cellular population, and interrogate their content. And their content comes from the cytoplasm of cells of origin that are subject to different stresses, to different disease processes, and on the other hand, may also be subject to the effects of a drug. And so if we have these exosomes isolated successfully, and then we interrogate them, all of a sudden we're able now to look, to open a window inside the cell and study the effects of the pathogenic mechanisms of disease, but also the effects of the drug on key cascades. This is something that it is an ongoing project, and we are, for this particular study, we've undertaken with Tom and Nigel's collaboration, and we're trying to look at key pathways in the insulin cascade in exosomes from patients in this clinical study, and we're hopeful that we will be able to have results out in the published fairly soon. But taking a step back, these are the particular biomarkers that we are interested in, and we think that they have the potential of revolutionize our ability to answer, to ask questions even, and to answer them in clinical trials in neurodegenerative diseases. There are other biomarkers that are more close to the clinical outcome that could be useful for showing neuroprotection. For instance, something like brain atrophy, which could be relevant to the cognitive effects of a drug and whether a drug preserves cognition. Well, for that, it may be more relevant to ask the question, is there increased brain atrophy? And this is something that you would be able to follow with MRI, looking at areas of the brain that are involved in cognition, or even a PET scan. That PET scan could answer the question about the functional ability of remaining dopaminergic cells. And so we have a particular angle in the biomarkers field, which I think it's uniquely powerful to answer certain kinds of mechanistic questions, but a variety of biomarkers likely will have to be involved in the design of a clinical trial, hoping to answer definitively variety of clinical effects and their underlying mechanisms.
So could I ask, when it comes to these blood-borne vesicles that are derived from brain, the exosomes, how are you certain that they come from the brain? Can you just give a layman's version to describe that, you know, these are from the brain and not from the liver or the kidney or the heart?
So, well, certain is a very strong word for us.
A good word if you're doing science.
For emerging field. We can have reasonable degrees of certainty on the basis of knowing that exosomes from the brain can cross the blood-brain barrier, and there has been pretty good evidence for that. And also, we find the population of extracellular vesicles that express multiple neuronal markers, and also at the same time another population that expresses multiple astrocytic markers that we also think that would be eventually useful in neurodegenerative disease. And well, we can ask, where did this population come from? I mean, it has to have come from somewhere. As of to the certainty of having the definitive certainty of knowing that this particular vesicle came from this particular neuron and this particular brain area, I would say the field is not there yet, but it is definitely a target for ongoing investigation.
So you hinted that you're in the process still of working on this project, but you also had some results. Can you share a little bit of preliminary information? Can you track in the exosomes changes that might be related to exenatide in the patients that Tom treated?
Well, with the caveat that they're still not have gone through the whole peer review publication to be widely accepted by other scientists other than me and Tom. With that said, I would have to say that we found evidence in these extracellular vesicles that the insulin signaling cascade was engaged, and we saw some markers up in the whole cascade, close to the origin of the cascade, in a molecule called insulin receptor substrate, but also downstream to it, further down in the cascade of events in the AKT signaling pathway, and further down in the mTOR signaling pathway. We saw coherent changes, and I would emphasize the word coherent because it was not a molecule going up and another one going down and who knows what it means, but it was what you would actually expect if you were having cells in a petri dish and you were conducting an experiment exposing them to an insulin sensitizing drug under really tight control conditions. You would expect, well, if this is a cascade, molecular events would go one particular way, all the on switches would be turned on and all the off would be turned off. And it was fairly similar to what you would expect to see there. And so the totality of evidence and the multiple markers that we got make me convinced that more than anyone in particular that we, in fact, were able with exenatide to engage the insulin cascade in neurons that generated these extracellular vesicles. So we're very hopeful that this upcoming paper will introduce this concept of following disease response mechanisms with extracellular vesicles will be something actually that other future studies will be able to take advantage of.
So at this point, I want to tell all the attendees we have about 15 minutes of this webinar left, and we won't forget your questions and we won't forget discussing what the future is for exenatide. But before we go there, maybe it's worth tying back to what Nigel said. Parkinson's disease is not the same in all patients, and it would seem that maybe some patients have more of one disease mechanism than another. So perhaps some patients have more issues with glucose metabolism than others. Tom and Demetrius, do you think that if you use biomarkers like this, it might be possible to identify people who might be more likely to respond positively to a drug like exenatide? So this is what we call personalized medicine or precision medicine. You pick the ones who really are most suited for that therapy, knowing that others who have the same symptoms of Parkinson's may actually need a different type of therapy. What do you think about this? How much variability was there in the original study, Tom, first?
So if we're looking at exosome biomarkers, then what's emerging is this change in the insulin resistance. And perhaps, you know, we haven't done this formally, but perhaps the analysis where someone who's got little insulin resistance at the start might predict that there'll be little response to exenatide, whereas someone that has a lot of insulin resistance should have a big response to exenatide. That would be a good hypothesis to test. We haven't done that. We've just looked at the overall insulin resistance in the group and see if the people on exenatide had a bigger improvement in their insulin resistance than the people on placebo. And that's what we have found. But we haven't taken the next step to see if we can use it as a predictor of response as yet. But it's something that deserves further explanation. But it's a bit of a post hoc.
So I keep on seeing Penny's picture on my screen here. I don't know if it's because you have a question in mind or if it's the seagulls that are in the background making noise.
No. The only comment I would have to say is that exactly what Tom said just before, or you said, I think, just before, Parkinson's is not one disease. Mm-hmm. It's a disease of many, many aspects, and no two people have the same. So I'm sure that it's relevant to establish which patients will benefit from certain drugs before experimenting with them even. And it's really interesting to hear this conversation. So
if you think back of the conversation just now, there are other things that have come up in your mind that as a participant you weren't aware of when you went into the trial? No, I think I was pretty well informed. I think I felt very involved, and I don't think there was anything that I've really learnt particularly new. I'm just grateful for the opportunity to have taken part in it, and I hope I contributed something towards the end results in due course. You have, definitely, certainly have. So I think it's a good time then to switch to the future. So let's talk about what is the future, first of all, for exenatide in Parkinson's disease. What will be the next trial? Will there be a next trial? How and where, if so? And secondly, we've had these questions that come in saying, what about other diseases like multiple system atrophy? So I'd like to start with the first question, and maybe, Tom, you're the best suited one for this.
Yeah.
Will there be a trial? How and when?
So where we are now is that we've shown that we've had a statistically significant, definite positive outcome in 62 people with Parkinson's disease. And you know, this is a fantastic level of evidence, but it's only in 62 people. So this needs to be reproduced in a bigger trial before any of us can really have the confidence that this is going to be a future treatment for Parkinson's patients. So what we're doing at the moment is we're planning a bigger trial that's going to be of 200 patients. We're going to do it in the UK, and we're hopefully on the last stages of getting the funding formally approved for that trial. And then when we have all the paperwork in place, we'll be recruiting to that trial in early 2019. What we're going to do slightly differently this time is, rather than have people exposed to either drug or placebo just for 1 year, we'll see them just as regularly, and at the end of 1 year, we'll see if there is the same effect size we've seen in the last trial, but we'll then carry on, and we'll see if the effect size at 2 years is larger than the effect size at 1 year. And if we do find that on placebo, things continue to deteriorate, but on exenatide, things continue to remain stable, or the two lines separate, then this really does conclusively demonstrate that there is a disease-modifying effect, and the cumulative advantage means it's something that people should be using on a much bigger scale in the long term. So that's what's planned. I can talk to you a bit about MSA as well. You know,
maybe I could say there's another question that just came in. Have all the people with Parkinson's lived in the UK who've been on these trials, or will you do trials in North America? That might be a simple answer.
So I can't do trials in North America, but I'm very happy to help colleagues who wish to.
And I could interject that there might be other agonists that target the same receptor, this GLP-1 receptor, that can be tested in North America. And there are also some trials already going on with drugs that are related to exenatide. So stay posted on the Cure Parkinson's Trust website, or read articles in Journal of Parkinson's Disease about this. But please go ahead then, Tom, and talk about MSA. And we heard it's a disease where the protein synuclein aggregates, but it is also a disease where it aggregates in a different cell type in the brain. It's mostly the cells that form the insulating sheaths along the long axonal connections that are affected in MSA. It's also a disease which typically is much, much more aggressive than Parkinson's disease. But tell us about trials here.
Absolutely. So because it's more aggressive, and because the response to dopaminergic drugs is so much more disappointing, if anything, the urgency and the pressing need to get a drug that may help slow down disease progression in MSA is even higher than it is in Parkinson's disease. So we are putting together a costing for an application to do a small, if you like, similar to the open label trial that we did with exenatide and PD originally, to do a small study in MSA patients as well. I do have anecdotal feedback from 1 or 2 patients with MSA who have tried exenatide, and I know that it's well tolerated. And we need to see if the effects we've seen in Parkinson's have any relationship to the effects we hope to see in MSA. So, you know, we're working as hard as we can to try and answer that question.
And do you know anything about diseases that not necessarily have the same molecular pathology, but affect basal ganglia, like corticobasal degeneration, progressive supranuclear palsy? Anything on there?
So, of course, we know MSA, there's insulin resistance from the papers that Nigel and Dimitros were referring to. And if that insulin resistance is the key, then MSA should respond in the same way as Parkinson's should respond. From PSP and CBD, there's no evidence of insulin resistance as yet. There's a different protein involved in the toxicity in those diseases compared to the alpha-synuclein in Parkinson's disease and MSA. But both of them, there may be a role of inflammation. And if inflammation is the critical mechanistic target of exenatide that the Ted Dawson paper alluded to, then perhaps there is a signal that these diseases may respond. So understanding the mechanism will help tease out which of these diseases to target next.
When it comes to your next trial in Parkinson's disease, since we have a few minutes left, I'm going to just try to understand how will you deal with this issue of the patients in the placebo, the quote-unquote untreated arm versus the exenatide active arm, not necessarily coming in at the same level at the start of the trial, because some of you may know this has been hotly debated in the neurology community that by chance they were randomized, they weren't at exactly the same levels. I don't think they were statistically significantly different when they started. But how will you handle this going forward and avoiding that situation?
So this is the problem in a small trial. If you only have 62 patients, then by chance there's possibility that the groups may differ at baseline. In a larger trial, that likelihood shrinks and shrinks and shrinks just because the randomization process becomes better. What we did in the first trial, we had to decide whether we should stratify patients according to their UPDRS scores at the start, or if we should adjust for the UPDRS scores at the start when we compare them at the end. And the statistical advice was that it's far better to do an adjustment at the end for any baseline differences rather than stratifying at the start. So that's what we did. So our comparison of the differences in severity at the end of the trial were all adjusted for any baseline differences. So the fact that they were different at the start doesn't make any worrying or alarming signal in how robust the analysis is at the end. So I'm quite confident in that.
So another question that came in now via the web, and I think it's an important question. We've talked all about the excitement around exenatide. What are the potential downsides, and what were the adverse events? Are there other adverse events that you are worried about if you treat large numbers of people for, not for a year, but for 10, 20 years, and especially aging people, because Parkinson's is still a disease which primarily affects older people?
Okay, so weight loss is the most common adverse event of exenatide. Now, in the diabetes population, where generally people are overweight, that's a good thing. In the Parkinson's population, there are many people that are already underweight, and weight loss will be an issue. From anecdotal experience and also from the trial data, we think that weight loss tends to settle over the first months, if not year of the trial, and ongoing weight loss tends not to be an issue. But in any population that might have swallowing problems or already on a downwards trajectory, this will be a concern. Other things are gastrointestinal upset, nausea, abdominal bloating. These are real issues, and whether there's any impact on medication absorption, levodopa absorption, that can be an issue. That was more of an issue in our first trial where we used Byetta, which is the twice daily injection, and far less of an issue when we use the once weekly Bydureon injection. But it's something that we need to counsel people about and look out for. In the long term, we need to be aware of associations between exenatide and other GLP-1 receptor agonists and problems with the pancreas. Now, there have been reports of acute pancreatitis, chronic pancreatitis, and pancreatic cancer. Now, in the very biggest observational studies of diabetes patients, there hasn't been any significant association between any of these illnesses and long-term GLP-1 receptor agonist use. But there's not without controversy here. And so we need to be the first to be the most vigilant about any changes in pancreatic enzymes or pancreatic function, such that if the signal is there, but only very small, we can spot it and make sure we don't do any harm to patients.
So just to be devil's advocate, we're all neuroscientists on this panel. What if the effect of exenatide in Parkinson's disease is primarily on the gut microbiome? Actually, it's a question that was emailed to us yesterday. We are learning that the gut microbiome might play a role in the triggering of the disease and potentially in the level of some of the Parkinson's symptoms. Could you conceivably imagine that exenatide is working primarily in the gut, changing the bacteria in the gut, and thereby as a secondary effect, changing the brain?
Well, it's entirely possible. So, because people do get GI symptoms as a consequence use, then it's entirely conceivable that the microbiome does change in a nuanced way as a result of this. It's a completely speculative area. We know that the microbiome is different in patients with Parkinson's disease compared to people without Parkinson's. And we know that in animal models of Parkinson's, you can influence neurodegeneration by affecting the gut microbiome with antibiotics or germ-free feed in alpha-synuclein mice. So we know that these steps are tantalizing to have this speculation. But in terms of evidence, we don't have the evidence to inform us robustly on that.
So I got an exciting notification from Helen Matthews at Cure Parkinson's Trust that we might be cut off in exactly 1 minute. So for that reason, I think we should wrap up. And I'd like to thank all the panelists. I'd like to thank all the attendees. We've had lots of questions come in. We didn't answer your question now. Maybe we can by email. And I'd also like to announce that we will have these webinars occurring quarterly. The next one will be sometime in September, the end of September, around the 26th, 27th, when we have the Grand Challenges and Rallying to the Challenges meeting here in Grand Rapids. And also I'd like to let you all know that this particular webinar has been recorded and it will be available on the Cure Parkinson's Trust, Journal of Parkinson's Disease, and Parkinson's Movement websites. And I think we are now approaching the last few seconds on my watch. So if we suddenly disappear, I'd like to say bye-bye and thank you once again.
Thanks very much and well done for sharing, Patrick.
Thank you.